In
this current state of innovation, scientists have gained the ability to edit, test, and modify the makeup of the human genome. With the invention of recent technologies like CRISPR, something previously believed as impossible has taken center stage over the course of historical alteration. Even so, as its evaluation has continued through its use and study, various concerns and complications have surrounded CRISPR's original intentions. In short, it's brought a multitude of questions towards its limitations. For instance, where is the line to how far some scientists will pursue this tool's path? What would it take to twist the intended meaning of its operations? Creation, and how long until the basis of CRISPR itself is no longer just a matter of gene alteration? From what is known, the facts and responses are significantly intricate, and with so many viewpoints, a variety of controversies are left to include and comprehend as well. However, with implementing both social-cultural and futuristic vantage points, it seems like these inquiries may find their counterpart. But as far as research can tell, this scientific tool is simply not ready. First observed in the late 1900s, genome modification technologies have been, upon recently, further developed on a much larger, highly advanced scale. The newest form of these technologies, the clustered regularly interspaced short palindromic repeats, or CRISPR process, immediately brought a variety of improvements to its predecessors. When introduced in 2012, its name bears reference to the one-of-a-kind sorting of brief, moderately palindromic sequences of DNA in both genomes of bacteria and microorganisms. The process involves the utilization of cellular defense mechanisms to penetrate and modify strands of genetic code infected with viruses. Being quicker, cheaper, and more elementary, its evaluation has shown to be significantly more accurate. With further use and study, its experimentation has not only led to focusing on inherited disorders, but on genes that give rise to specific diseases such as cancer and heart-related illnesses. However, over the past 3 decades, the overall support of gene alteration has followed in a decreasing inclination. Why is this? Well, from a societal standpoint, the entire process of gene editing can influence and fluctuate an entire generation. Regardless of this, it's clear that whatever their possible impact, one would think that a larger discussion of CRISPR will be held in advance of a complete release to the public. However, it wasn't until 2015 that a call for a broad societal consensus was issued, and even then, questions still remain on how it could reach full inclusivity and participation from the global community. With a worldly observation of gene editing type approach, the inclusion of the human population is essential for a deeper and thorough explanation of what the procedure of gene editing means in all perspectives. Although due to this being such a broad, revolutionary, and relatively new process, there is a multitude of documentary and reported discourse in terms of opinions. But this should be expected, as CRISPR is still in its very early stages of development. To better understand these viewpoints, the United States in particular created a variety of public polls on both the web itself and social media platforms. In each area, the research tracked the current and most popular trends when it came to opinions on CRISPR. Whether positive or negative, both delved into the where and what was being expressed and to what degree. In its conclusion, various fears and concerns appear to be more common than originally thought. The fact that CRISPR-Cas9 has and will continue to be tested on human embryos resulted in alarm for its usage towards designer babies. This would bring about the reality of humanity being able to alter and assign specific or even desirable traits and talents to later generations. What does this tell us about the global community about the future of CRISPR technologies? How is possessing the power to alter human evolution going to keep itself from converting negatively? Even now, these cards are beginning to unfold. Upon recent news, it has been reported that CRISPR can cause grave reactions in cells found in human embryos. This is, in statement, giving rise to nuclei rejecting large pieces of their genetic material. Some separate from the previous cells were so confused by their modifications that they renounced their attempts to fix them. This resulted in the disposal of entire chromosomes. So for the future of human beings to be rearranged in terms of DNA sequence, one miscalculation can be irreversible and gravely fatal to those following. It should be stated that there are and have been many successes when it comes to the creation of CRISPR-Cas9. However, that doesn't mean that the world can't prepare for the negative effects in which weren't intended. It wasn't until a variety of instances that these consequences became shockingly apparent. As previously introduced, one in particular implemented a legal infant modification. In 2018, a Chinese scientist used gene editing technology to create the first modified twin infants. After they became known to the public, the experiment was sentenced as highly dangerous and reckless. This just comes to show how much we have yet to understand about ourselves. Another example, in 2017, a group of researchers attempted to use CRISPR-Cas9 to break apart a copy of a healthy gene to repair another containing a mutation. At first, the lab was announced to be a success. Yet, half the embryos that were used went at a loss to manage the damage caused by the rip. In short, doctors claim that there is a long road of improvement still lies ahead. It is not until scientists figure out the roots of the gene altering results that CRISPR in its entirety will remain harmful to future generations.
Very good. I wasn't quite sure. How did your research question evolve as you moved through the process?
To be quite honest, from the beginning, I was undecided whether or not I wanted to support gene editing or not. I did know some positives and drawbacks from each argument. However, researching helped me to pursue what each side was imposing. But really, I expected myself to move in the direction of against, since the consequences only seemed to worsen for CRISPR as I delved deeper.
Okay, thank you. Another question. How was your conclusion in conversation with the body of literature or other research sources that you examined?
What was that again?
So, how is your conclusion in conversation with what people are thinking and the research that you've done so far?
In honest response to my examples on previous gene editing tests, for all I included, I made it clear that CRISPR is simply too dangerous to be incorporated into human beings at the moment. There is simply too much we don't know about what could happen if a miscalculation is made, and conclusively, my conclusion exerts the facts with all seriousness.
Okay, thank you. All right, another round of applause.